In the early 1950s, scientists knew DNA carried heredity, yet nobody knew what it looked like. The letters of life were in their hands, but the page was unreadable. Then a photograph appeared. At King's College London, Rosalind Franklin fired X-rays through DNA and captured an image labeled Photo 51, a pattern that pointed to the answer. And soon, without her knowledge, that image reached Cambridge, and everything changed. Within weeks, Watson and Crick built the double helix. But the story didn't end with a discovery. It began a controversy. Who found the secret of life, and who paid the price? In this video, we'll follow the race, the photograph, and the credit battle that still haunts the most famous structure in science. Imagine trying to solve the greatest puzzle in biology while missing the picture on the front of the box. By the early 1950s, scientists had strong evidence that DNA, not protein, carried genetic information. In 1944, Oswald Avery and his colleagues pointed to DNA as the transforming principle, and in 1952, the Hershey-Chase-Phage experiment pushed the field even further toward DNA as the genetic material. But here's the problem. Knowing DNA the code is not the same as knowing how the code works. To understand heredity, scientists needed one thing above all: DNA's three-dimensional structure. Because whatever shape DNA had, it had to explain one thing: replication. And there were clues. One of the biggest came from a chemist named Erwin Chargaff, who found a weird, stubborn pattern in DNA. A and T appear in equal amounts, and so do G and C. Those ratios didn't explain the shape by themselves, but they sounded like the rules of a lock, waiting for the right key. Now the race was on, and it had three main fronts, each with a totally different style of genius. In California, Linus Pauling had already become a legend by figuring out the alpha helix in proteins. He was brilliant, fast, confident, and he believed the DNA problem was his to win. In early 1953, Pauling published a triple helix proposal for nucleic acids. A bold swing that turned out to be chemically wrong. But even being wrong was dangerous, because it proved the finish line was close. Pauling wasn't just competition, he was a countdown clock. At King's College London, the work looked nothing like Pauling's sweeping theorizing. It was slow, technical, unforgiving. This was X-ray diffraction. Not a picture, but a pattern, data you have to translate into structure. Franklin worked inside the MRC biophysics unit at King's, run by Sir John Randall, and she focused on making DNA fibers good enough to speak clearly through X-rays. And with her student Raymond Gosling, she produced an image so clean it would become legendary. Photograph 51. One basement, one photo. A clue powerful enough to end the mystery if the right eyes saw it. Meanwhile, in Cambridge at the Cavendish Laboratory, James Watson and Francis Crick were taking the most audacious approach of all. They weren't primarily running DNA experiments. They were trying to build DNA, physically, as a model that had to obey chemistry and match the data. Their first attempt was a disaster. After presenting a wrong model, they were effectively told to back off. Lawrence Bragg, head of the Cavendish, ordered them to stop working on DNA and leave it to King's. But obsession doesn't obey orders. By early 1953, the pressure spiked again. Pauling was closing in. King's College had the best images, and Watson and Crick were desperate for the one thing they lacked: the decisive piece of structural evidence. And that's what makes this next part so tense. Because the race to decode life wasn't going to be won by the biggest lab, or the most famous scientist, or even the most careful measurements. It was going to be tipped by a single image, clear enough to change history, and controversial enough to haunt it. Rosalind Franklin didn't enter the DNA story as a side character. She entered it as the person who could actually extract truth from a blur. By 1950, Franklin had already become exceptionally skilled at X-ray diffraction, especially after her research years in Paris, where she trained under Jacques Meringue and mastered the craft of turning diffraction patterns into real physical structure. That's why Sir John Randall recruited her to King's College London. Depending on the source, she was recruited in late 1950 and began working there in early 1951, right as the DNA race was heating up. But the King's lab came with a hidden trap. Nobody clarified the chain of command. Randall had privately told Franklin she would be in sole charge of the DNA diffraction work, without making that clear to Maurice Wilkins, who had already been working on DNA and assumed they'd be partners. That single miscommunication turned a collaboration into a Cold War. Still, Franklin didn't argue her case with speeches. She argued it with data. She worked where DNA actually lived, not in chalkboards or theories, but in fibers, humidity levels, sealed cameras, and long exposures. She proved DNA could exist in two forms depending on water content, an A form and a more hydrated B form, meaning the molecule could literally change its diffraction signature if you treated it slightly differently. And with a graduate student, Raymond Gosling, she began taking images that were so clean, so precise. They didn't just suggest an answer, they forced one. Here's the part that feels like fiction, but isn't. In May 1952, Franklin and Gosling set up a DNA fiber experiment under high humidity conditions and ran an X-ray exposure for 62 hours. The film was developed days later. It was simply labeled by its number in the series, photograph 51. And when you look at it, you can see why it became legendary. It isn't a picture of DNA. X-ray diffraction doesn't give you that. It gives you a pattern, spots, and smears produced when X-rays scatter from repeating atomic structure. But in photo 51, that pattern forms a bold, unmistakable X shape, the classic fingerprint of a helix. Even better, it wasn't just helix vibes. The spacing and symmetry in the pattern contained hard geometry, enough to infer key features like the regular stacking inside the helix and the approximate repeat structure, the rhythm of the molecule. Franklin understood the power of what she had, but her style was the opposite of a gambler's. She didn't want a headline, she wanted certainty. According to King's own account, she returned to this data in early 1953 and drafted a write-up stating a helical structure was highly probable, likely a double helix, and she had already inferred crucial structural details. And then the story takes its sharpest turn. Photo 51 would soon be shown to someone who wasn't supposed to see it. The photo didn't solve DNA by itself, but it moved the finish line into view, and it poured gasoline on the final sprint. Next, we'll follow what happens when that one image collides with model building ambition in Cambridge, and how, within weeks, the double helix goes from mystery to the most famous structure in science. While Franklin was building her case the hard way, one exposure, one measurement, one calculation at a time, another front of the DNA war was burning hot. At Cambridge, James Watson and Francis Crick were doing something almost audacious for biology in 1951. They were trying to build DNA before fully understanding it. No giant X-ray cameras, no humidified DNA fibers, just model making, cardboard bases, metal rods, and a stubborn belief that if the structure was real, it would click into place like a lock finding its key. Their method was fast, creative, and dangerously vulnerable to being wrong. And they were wrong. After Watson attended a seminar where Franklin presented her X-ray results, Watson and Crick rushed into their first serious attempt, a model that looked plausible on a table, but collapsed under chemistry and Franklin's critique. The reaction was immediate and humiliating. King's College complained they were stepping on toes. The model was judged a mess. and the head of the Cavendish Laboratory, Sir Lawrence Bragg, ordered Watson and Crick to stop working on DNA and leave the problem to King's. So now the race split into two wildly different styles. King's College. Slow, disciplined, experimental precision. Franklin separating DNA into A form and B form by humidity and water content, refusing to guess until the data forced the answer. Cambridge. Restless theory, model building instinct, and two men who had just been benched, but were still staring at DNA like it was a safe they knew could be cracked. For a while, it looked like King's had the evidence, and Cambridge had the ambition. Neither side had everything. And then, the rivalry stopped being a rivalry and became a turning point in scientific history. January 1953. King's College, London. Maurice Wilkins calls James Watson in quietly. Not a public seminar, not an official data exchange. Just a moment in a room that would later echo through every biology textbook on Earth. Wilkins holds up a photographic plate to the light. Photo 51. Franklin isn't there, and she doesn't know it's being shown. Watson looked for only a few seconds, but that was enough. He didn't need a lecture. He already knew what he was seeing. Watson later wrote that the moment hit him instantly, like something clicking into place. And that's why this moment is still uncomfortable today, because it raises a question bigger than DNA. Was this scientific collaboration, or was it the quiet transfer of someone else's decisive evidence without their knowledge? King's notes that Franklin was unaware the photograph had been shown to Watson, and that Wilkins assumed Watson had already seen earlier helix cross images. Yet photo 51 was dramatically clearer than anything before it. One photograph, a few seconds, a lifetime of consequences. Because once Watson walked out of that room, the benching at Cambridge didn't matter anymore. The final sprint had begun. Once Watson saw photo 51, the DNA mystery stopped being foggy philosophy and became something sharper, a problem with dimensions. Back in Cambridge, the Cavendish Laboratory turned into a workshop. Watson and Crick weren't just imagining DNA anymore. They were trying to build a structure that couldn't cheat. It had to match the X-ray geometry, obey chemistry, and explain the one miracle of heredity: how DNA copies itself. Now they had constraints, enough to build instead of guess. Then they brought in the clue that had been sitting in plain sight for years: Chargaff's rules. A appears in the same amount as T, and G in the same amount as C. That wasn't a coincidence. It was a constraint, a lock. Suddenly the question became almost cinematic. What pairing keeps the helix the same width every step, all the way up? Only one answer worked cleanly. A pairs with TG pairs with C. Two specific matches, held together by hydrogen bonds, fit like puzzle pieces across the center, keeping the helix stable and uniform. And here's the part that made the structure feel like destiny. If each strand carries the information to build its partner, then DNA doesn't just store a code. It explains replication. Split the molecule down the middle, and each half becomes a template. Life copying itself suddenly looked not mysterious, but mechanical. By March 1953, the double helix was standing in the Cavendish, metal plates and rods forming a staircase that twisted upward like a revelation. And that's when the story hits its most famous scene. Crick walked into the Eagle Pub and, according to multiple accounts, announced something like, "We have discovered the secret of life," not because they'd found a poetic metaphor, but because they'd found a structure that made heredity work. The world didn't learn the secret of life through a dramatic press conference. It learned it through ink. Nature published 3 papers in the same issue, back to back, like 3 pieces of a single explosion: Watson and Crick, the double helix model; Wilkins and colleagues, supporting diffraction evidence; Franklin and Gosling, the crucial X-ray analysis and measurements that made the model physically real. 3 papers, 1 discovery, and a scientific earthquake that would reshape medicine, genetics, and every biological field that followed. But the triumph carried a shadow, because once the model was solved, a different question began to grow, quietly but relentlessly. Who actually deserved the credit for cracking DNA? The Nobel Prize in Physiology or Medicine went to Watson Crick and Maurice Wilkins for the discovery of DNA's structure and its significance for information transfer. 3 names became the official story, but one absence was impossible to ignore: Rosalind Franklin. Franklin had died in 1958 at just 37 from ovarian cancer, so her name could never appear on that stage. No speech, no medal, no global recognition in the moment it mattered most. And then, years later, the public story hardened, largely because of one book. Watson published The Double Helix, a memoir that became famous not only for the discovery, but for how it portrayed Franklin. Often dismissive, personal, and unflattering, more character sketch than scientific tribute. That book shaped how generations imagined her, not as a central mind in the discovery, but as a difficult obstacle in the way of genius. But history doesn't stay frozen. As scientists and historians revisited notebooks, correspondence, and King's College records, the picture sharpened. Franklin wasn't a helper. She was producing the best structural DNA evidence in the world, distinguishing DNA forms, controlling experimental conditions with rare skill, and extracting the geometry the model depended on. And crucially, the modern discussion isn't only, did they steal? Because the culture of data sharing was messier and more informal than today, and documents suggest multiple pathways by which Cambridge gained access to King's results. The real, lasting point is simpler and heavier. Franklin's work was essential, and for a long time, the public story didn't treat it that way. Today, she's increasingly recognized as what she always was, not a footnote, not a side character, but one of the architects of the most important biological structure ever found. And that's what makes the DNA story unforgettable, because it isn't just about molecules. It's about ambition, rivalry, fragile ethics, and the strange way truth can be discovered while credit slips through human hands. Photo 51 didn't look like a revolution. No colorful diagram, no microscope snapshot, just a ghostly X on a strip of film, light and shadow in a London lab, stored away while the analysis continued. But that X helped make visible something humanity had never truly understood before: the mechanism of heredity. Once the double helix was understood, life stopped being a mystery you could only describe and became something you could test and predict. Genetic information wasn't floating in cells like fog. It was written in a sequence. And because the bases pair, A with T, G with C, each strand carries the information to rebuild its partner. Unzip the helix, and each half becomes a template. Replication stops being magic. It becomes chemistry, and that single insight rewired medicine. First, disease became readable. Many illnesses could now be traced to errors in the code, mutations, miscopies, broken instructions. Cancer, especially, shifted from cellular chaos to something sharper, a disease driven by genetic changes that disrupt the cell's control systems. You don't just treat symptoms, you hunt the underlying error. Then, biology became practical. New tools let scientists read and copy DNA, sequencing and amplification, and biotech turned cells into factories. One landmark example, insulin. What used to come from animal organs could now be made by engineered microbes, medicine produced from genetic instructions. DNA even left the clinic. In forensics, a trace of blood or hair could identify a person, often with extraordinary precision, evidence speaking in the language of genetics. And the story kept accelerating. The Human Genome Project turned the idea of the instruction manual into a reference map. And today, With CRISPR, we've crossed a line that would have sounded like science fiction in 1953. We're not just reading the manual anymore, we're beginning to edit it. So yes, one image changed medicine forever, because Photo 51 wasn't just a clue to a shape. It was the moment humanity finally saw the architecture of inheritance and realized the future could be built on it. All of this began with patterns of light and shadow on a picture almost no one saw. So, if you want more origin stories where science feels like a human epic, full of rivalries, mistakes, breakthroughs, and consequences, subscribe and join the channel.